We could have our next speaker up. Our second talk today will be human iPS cell-derived pancreatic cells, correct hyperglycemia in diabetic mice, by Drs. Manzar, Kim, Roti, and Zavazava from Iowa City.
Thank you very much for the kind introduction. I have nothing to disclose. Today I'll be talking to you about skin cell-derived induced pluripotent stem cells derived from type 1 diabetic patients and how these cells can be differentiated into insulin-producing cells for the cure of type 1 diabetes. Just a little background about type 1 diabetes. It's an autoimmune disorder targeting pancreatic beta cells located in islets. And when insulin-producing pancreatic beta cells are destroyed, this results in insufficient insulin production, a buildup of glucose in the blood, which can cause a host of secondary complications, such as blindness, neuropathy, and kidney failure if left unmanaged. It's been estimated that over 25 million people worldwide have type 1 diabetes, and because it is progressively targeting younger individuals, decades worth of research has been devoted to finding a cure, which has unfortunately not yet emerged. Current management of type 1 diabetes largely relies on insulin replacement therapy, which has several disadvantages. First being that it's not physiological, it must be adjusted according to the circumstances the patient encounters, and if mismanaged, the consequences might be fatal. Additionally, these patients often succumb to the complications of type 1 diabetes. Clearly, there is a need to identify a better source for the cure of these patients, and one of that is the Edmonton protocol, in which cadaveric islets are isolated from various donors and pulled together before being transplanted into the patient. The disadvantage with this approach, the main one, is that there's a severe shortage of organ donors, which limits widespread application of this technique. Additionally, because the donors are allogeneic to the recipient, there is the need for immunosuppression, which exacerbates type 1 diabetes, and often these grafts are eventually rejected, requiring recurrent transplantation. And thus, there is a need to identify an alternative source of pancreatic beta cells, for which we believe are induced pluripotent stem cells. iPS cells are somatic cells reprogrammed into an embryonic stem cell fate and can differentiate into tissues of any germ layer. These cells were discovered by Dr. Yamanaka, and he won the Nobel Prize for his discovery 2 years ago. In the context of type 1 diabetes, in the context of using iPS cells for the cure of type 1 diabetes, the diabetic patient would donate skin fibroblasts, which upon being reprogrammed into induced pluripotent stem cells, would be differentiated into insulin-producing cells. These cells would then be transplanted back into the same patient, hopefully for long-term correction of hyperglycemia. There are two key advantages with this approach, first being that the theoretical availability of these cells is unlimited, and second, because these cells are derived from the patient and going back to the same patient, the patient-tailored application of this technique is precluding immunosuppression. So the goal is to go from the skin biopsy of the diabetic patient to the human islet, and how do we get from there to here? This has been a question that we've been addressing in our lab for several years, and I'm going to show you some data that we've accumulated. This is from several years ago. Here we have an iPS cell colony, which was converted into definitive endoderm cells, which is the first stage of differentiation. These cells are then differentiated into pancreatic precursor cells that coalesce into islet-like spheroids at the end of the differentiation. When these cells were transplanted under the kidney capsule of diabetic mice, they formed a white, fleshy, pancreatic-like tissue on top of the kidney, and H&E staining revealed that the morphology of this was highly distinct from the glomeruli containing kidney. Additionally, the presence of blood vessels suggested the ability of this tissue to attract and form vasculature. We were very enthusiastic by this data, but it was dimmed at the same time because the morphology of the structure seemed largely exocrine, meaning that these cells appeared to produce digestive enzymes instead of endocrine hormones. And this is supported by the notion that it took 100 days for the correction to manifest in the diabetic mice into normal glycemic or near normal glycemic range. We believe that these cells, although were insulin-producing cells when they were transplanted, were really immature and eventually reverted into an exocrine cell fate upon transplantation. And these aren't just problems that we've observed. It's been a long-standing trend in the field that the major problems with current protocols are the poor efficiency of differentiation and the fact that immature cells are generated, whereby they express multiple hormones and are not responsive to glucose. In order to resolve these shortcomings and allow for more efficient and rapid correction of hyperglycemia, our goal was to generate mature, monohormonal, and glucose-responsive insulin-producing cells in vitro. How do we introduce such drastic changes into the differentiation protocol? Well, we looked at the basic platform of differentiation and identified that most labs, including our own at the time, utilized two-dimensional differentiation, which is very distinct from the three-dimensional differentiation of embryonic stem cells in the inner cell mass. We hypothesized that more physiological three-dimensional differentiation would allow for cell-to-cell crosstalk and signaling that would better improve the efficiency and functionality of the differentiated cells. And so we seeded our stem cell clusters in Matrigel, a bioactive extracellular matrix concoction, on top of which we placed stage-specific differentiation media. We also optimized several stages of the differentiation protocol. I'm not going to go into the details because of time, but basically we drove the pluripotent stem cells through 5 different stages lasting 27 days. First, the stem cells are converted into definitive endoderm cells expressing CXCR4 and SOX17. Eventually, these cells become posterior foregut progenitors and pancreatic progenitors that express the pancreatic master regulator PDX1. These cells then become endocrine progenitors and finally insulin-producing cells. Here, we took iPS cells derived from a diabetic patient and differentiated them using this novel 3D culture optimized differentiation protocol. And you'll see that at the end of stage 1, over 90% of the cells express markers typical of definitive endoderm, CXCR4 and SOX17. These cells eventually differentiate into over 96% PDX1 expressing cells. PDX1 is the pancreatic master regulator, without which you don't get a pancreas. And so for us to see almost 96% of the cells expressing PDX1 was really exciting. Further downstream differentiation of these cells gives rise to approximately 50% of the cells expressing insulin, which is a frequency that is comparable to what we observe in human islets. This is also significantly higher than what has been reported in the literature, with about 10% of the cells expressing insulin, and demonstrates the superiority of our protocol in generating insulin-producing cells from pluripotent stem cells. These cells also express insulin as evidenced by immunofluorescence, and we also observed a formation of some larger organoids that strongly expressed insulin. Macroscopically, these clusters resembled islets, and when they were immersed in dithiazone, which is an organic compound that binds to zinc found in insulin granules, they stained red in a manner very similar to islets. We assessed the expression of other markers typical of pancreatic beta cells, such as C-peptide and NKX6.1. You can see that the expression of C-peptide, which is a byproduct of insulin processing, is very strong in our cells, and this suggests de novo synthesis of insulin. Nuclear expression of the pancreatic beta cell transcription factor NKX6.1 confirms a pancreatic beta cell identity of these cells. We examined the ultrastructure of our cells and compared them to that of human islets using transmission electron microscopy. And what you'll see here is the pancreatic beta cell within the islet containing pockets of granules with very distinct morphologies, representing different maturities. First, you have the early insulin granule, which is round and gray. Then you have the maturing insulin granule, which is dark and round. And finally, you have the most mature insulin granule, which is angular and dark. And what you'll observe is that in all 3 of these granule subsets, they're surrounded by a halo, kind of like a fried egg morphology, which is very specific and characteristic of the insulin granule. Our insulin-producing cells possess very similar granule morphologies, and enumeration of the number of granules between these 2 cell types reveals a non-statistically significant difference. The most important question of all is now, are these cells functional? Do they respond to high glucose stimulation with insulin secretion? And indeed, you'll see here that yes, they do. When these cells were subjected to high glucose stimulation, we observed profound insulin secretion, and the specific index revealed about 10-fold increase in insulin secretion after exposure to high glucose. We're currently in the process of transplanting these cells into mice, and so the data are still kind of preliminary, but we see promising results here with about correction in as early as 3 weeks post-transplantation. And so I hope that I've convinced you today that 3D differentiation allows for the conversion of skin cells into pluripotent stem cells that can differentiate into insulin-producing cells, which resemble pancreatic beta cells and are glucose responsive, and that these cells can be a source for the cure of type 1 diabetes as an alternative to cadaveric islets. I also hope I show you today that the iPS cells can be coaxed into becoming iPSCs that form vascularized organoids upon transplantation and correct hyperglycemia. I'd like to end with thanking my mentor, Dr. Zavazava, as well as other Zavazava lab members, and our funding sources and the IIDP for providing us with human islets. I'll be happy to take any questions at this point. Thank you.
Thank you. For a non-islet scientist, that's very impressive work. Are there any questions from the audience? Microphone 10 at the back.
Shapiro, New York. Very interesting presentation.
Thank you.
If this research matures and you start transplanting diabetic patients, what's to prevent autoimmune recurrence?
That is the million-dollar question, and I don't have a great answer to that at this point, but I have some hypotheses that I'd like to go over potentially. So immunological findings in type 1 diabetes denote that it's the CD8 T cells that kill the pancreatic beta cells expressing insulin on MHC class 1. And autoantibodies that mark type 1 diabetes are not pathogenic. We believe that if the pancreatic beta cells don't express MHC class 1, they might be able to evade autoimmune recognition and attack. Stem cell derived cells, as well as stem cells, are famously immunoprivileged, meaning that they don't express MHC class 1 or class 2. And we've shown that our insulin-producing cells, similar to their precursor iPS cells, don't express class 1 and class 2. And we worried about whether that would make them susceptible to NK cell killing, and we show here that these cells, very similar to the pluripotent stem cells, are not susceptible to NK cell killing despite poor expression of MHC class 1. This altogether suggests immunoprivilege of the insulin-producing cells, similar to what's been described for other cell types in the literature. Thank you.
Okay, I'd like to invite the next speaker.
Can you escape?
Yeah, just press escape. Just look up here. Don't press any keys.